Well, good morning. Welcome. My name is Margaret Sutherland. I'm a program director at the National Institute of Neurological Disorders and Stroke. I'm broadcasting from Washington, D.C., and we're going to talk about Huntington's disease over the last 20 years and counting is the title of the topic. The idea today is to really give you an idea of what the National Institutes of Health cover in terms of Huntington's disease research and highlight some of the significant breakthroughs that have happened over the last few years that are enlightening us in terms of mechanisms associated with this disease and hopefully directing us towards potential new therapies for Huntington's disease. So the just to give you an idea of who we are, the National Institutes of Health is made up of 27 institutes and centers. It basically has two goals. The first goal of the NIH is to support science in pursuit of fundamental knowledge about the nature and behavior of living systems, and then basically apply this knowledge to extend healthy life and reduce the burdens of illness and disability. NIH is the largest funder of biomedical research in the world. More than 80% of the NIH's budget goes to roughly research personnel at over 2,500 universities and research institutes. It also supports about 6,000 scientists who work in the intramural research laboratories at NIH, and many of these scientists are on the main campus. The main campus also has the NIH Clinical Center, which is the largest hospital in the world, which is totally dedicated to clinical research. Over the last century, NIH has supported roughly 144 Nobel laureates who have been involved in things like discovery of MRI, understanding how cholesterol is regulated, understanding how viruses may play a role in cancer, and many other breakthroughs in medical research. So for our institute, which is the National Institute of Neurological Disorders and Stroke, our mission is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. This year, January 17th, President Obama signed the Consolidated Appropriations Act, and this basically presented a budget for NINDS of about $1.5 billion. We really want to stress here at the Institute that we are committed to funding investigator-initiated basic research, and that basic research is involved in elucidating the structure and function of the nervous system, as well as supporting translational and clinical research that will help identify causes and treatments for hundreds of disorders that affect the brain and nervous system. This year, 2013 to 2014, NINDS has also supported some major planning efforts in areas like epilepsy, Alzheimer's disease-related dementias, and Parkinson's disease. And these planning efforts bring together leading scientists in the field, clinicians, basic scientists, as well as the patient community and nonprofit government organizations also working in these disease spaces to help set a stage for where we hope research and discoveries will head over the next 5 years. The other major impact NINDS has had over the last year is basically bringing together the research community, journals involved in neuroscience research publications to talk about how we can, as a community, improve the preclinical and clinical research study design and transparency in reporting so that data that are contributing to decisions in terms of what targets and what molecules to move forward for clinical research and therapy development has a solid foundation for those treatments that we hope to advance. To give you an idea, and anyone basically can go to the NIH site and look at Reporter to look at disease categories and find out how much NINDS or NIH is funding. In this case, we're looking at Huntington's disease, and for instance, in financial year 2013, we funded $55 million in Huntington research. And to give you a breakdown, this is across NIH, across all institutes that are funding Huntington's disease research. NINDS funded $32.98 million in research grants. Now, what is Huntington's disease? So basically, Huntington's disease is a chronic and progressive brain disorder that is often clinically manifest in the 3rd to 4th decade of life. Early signs and symptoms of Huntington's can include irritability, depression, small involuntary movements, poor coordination, and cognitive impairment. As the disease progresses, and this progression can occur over 10 to 15 years, the movement disorder becomes more pronounced. Huntington's is a monogenetic disorder, so it's caused by a pathogenic expansion of the CAG repeat sequence in exon 1 of the Huntington gene. This mutation was identified in 1993, but to date, no treatment is available to either slow or reverse the progression of this devastating neurological disorder. NIH investigators are pursuing basic, translational, and clinical research to understand pathways and therapeutic targets that will lead to improved treatments for Huntington disease. On the right side of the screen, you can see what a normal brain would look like. That's the image on the left. The image on the right basically shows you what a Huntington brain looks like, where you see enlargement of the ventricles and significant loss in both the cortical and striatal areas of the brain. The image underneath the two brain images is basically what happens during advancing Huntington's disease. And so you can image the brain using MRI and look for where you see losses in brain volume. And so stage 1, you see the red areas indicates where initial volume loss is identified. And then as you move through the, or as the disease progresses, you can see in stage 2 and stage 4 that you see significant increases in the losses both in cortical and striatal areas. So interestingly, Huntington disease and the awareness of Huntington's disease was at least in part brought to light through Woody Guthrie, who was a folk singer responsible for This Land Is Our Land. He was diagnosed with Huntington's disease and he died in 1967. Margaret Guthrie, his wife, established the Committee to Combat Huntington's Disease, which is now the Huntington's Disease Society of America. And the HDSA, or Huntington's Disease Society of America, is really a grassroots organization that is an advocacy group who brings awareness and tools to families and patients with Huntington's disease. Marjorie, after Woody's death, basically continued to work with NIH. A paper was published in 1967, a review article on Huntington's chorea. And then she continued to work with NIH up until her death in 1983. And at that time, NIH instituted the annual Marjorie Guthrie Lecture in Genetics. Another major contributor to raising the awareness of what Huntington's disease is were the Wexlers. So in 1968, the Wexlers took on HD after his wife, Milton's wife, was diagnosed with Huntington's disease, and Nancy and Alice's mother's name was Leonore. So Milton established the California branch of CCHD, which was focused on basic research. The branch soon became the autonomous Huntington's Disease Foundation, and HDF has pioneered HD research and worked with NIH to advance our knowledge of what Huntington's disease is. Last year, Nancy Wexler published a paper in the Annual Review of Medicine, which, if you don't know much about Huntington's, is a great article describing what her mother went through during the course of Huntington's disease, and really how they found out about it at that time. And the time they found out about it was before the gene had actually been cloned. So one of the statements in the paper, and this is Nancy speaking, I was 23 years old and my sister Alice was 26 when our father Milton Wexler told us that our mother Leonore had just been diagnosed with a fatal brain disorder called Huntington's disease. Through support from the HDF, Nancy and a group of scientists went to Venezuela, where there was a large cohort of Huntington families around Lake Maracaibo. And through clinical research and building of what you see on the right-hand side, extensive pedigree or family histories of the disease, this was in part what contributed to the initial identification of the polymorphic DNA marker that linked to Huntington's disease. So this work was published in 1983. Jim Gazzella was the first author on the paper. The pedigree you see underneath the title for the paper was an American family that was brought together through efforts that NIH funded for an HD roster. Using this pedigree plus Venezuelan information, they were able to identify at least where the Huntington gene was located, and it was located on chromosome 4. Now, back in 1983, they didn't have all the tools we have today for DNA sequencing and mapping of genes. So it was a considerable effort and required a lot of technology development to really move along that chromosome and identify what the actual gene was. And a large number of groups were involved in this research, 6 in all, who came together in 1990 to build a consortium where they felt that it would be faster for the 6 groups to work together than work independently. And so this consortium was tracked in terms of advances. They came together in 1990. In 1992, a science paper came out talking about the Huntington gene quest goes on. And then in 1993, in a Cell paper, the breakthrough came where the mutation in the gene was identified by Jim Gazzella's group. And actually, it was really a work of the 6 groups all together and the consortium that was developed. So the paper that talks about this gene discovery was published in Cell, as I said, in 1993. And it was supported through NIH and the HDF and other organizations. And I think the important thing here and what makes Huntington unique is that it was really this collaboration that helped to move HD research forward. And so very unique at that time was the fact that the authors on the paper were the Huntington Disease Collaboration Research Group. So what do we fund today, and what are some of the grants that fit under these different categories? So again, to emphasize, NINDS funds a broad spectrum of Huntington's disease research. We are interested in basic research in terms of mechanisms, targets, and circuitry that both are related to the Huntington protein and processes that the Huntington, cellular processes that the Huntington protein plays a role in. So examples of these types of basic research grants are ones from Dimitri Krantz that's looking at protein acetylation and selective autophagy and how that plays a role in regulating Huntington protein homeostasis. Another very interesting and fairly recent finding is how that Huntington protein may play a role in striatal neurogenesis. For translational research, we're interested not only in supporting research that will help identify small molecules or biologics that can be used as therapeutics for Huntington's disease, but also in clinical research that will tell us more about the human biology of the disease. So an example of the small molecule approach is a grant funded under the NINDS Translational Research Program, and it's focused on novel histone deacetylase inhibitors. And the principal investigator on this grant is Joel Gottesfeld, and it started out as a collaboration between this group of investigators and Repligen, and now Repligen has transitioned that to BioMarin. The example of how we fund clinical research is research, for instance, that Stephen Rao's doing, looking at functional connectivity and pre-manifest Huntington's disease. The unique opportunity for Huntington's disease is the fact that we know the gene mutation, so we can identify individuals who study those individuals before you actually see manifestation of the disease in terms of a motor manifestation. In terms of human clinical therapeutic trials, we fund clinical trials that will move small molecules forward for therapeutic development, but we also fund research that helps us to develop better tools for Huntington's disease. In this case, this is an example of a grant that is looking at different ways to assess the quality of life measure in Huntington's disease. And NIH has developed a toolbox, and this grant is testing out several of those clinical assessments to see if they're better or more sensitive markers of that quality of life measure. So it's obviously been a great advancement to identify the gene, but what can NINDS do in terms of helping to further transform HD research? And what might those fundamental principles be? So what are the key principles that will change the field and really advance the field, accelerate how we look at Huntington's disease, is through broad data and resource sharing. And I'm going to give you an example of that resource and data sharing in the next couple of slides. Another principle that we've applied over the last few years is to partner to accelerate discovery. And partnering occurs not just by bringing groups of scientists together who have different expertise, but also to partner with non-government organizations and industry to find ways to advance research in where we couldn't do it as a single entity. We've also worked with the research community to develop best practice guidelines to improve transparency, control of bias, and scientific rigor. And we feel this is incredibly important for really helping to move the target validation we're doing around different potential new therapeutic moieties for Huntington's disease, and hopefully improving the preclinical efficacy of those studies. This really fits in with the rigor guidelines that NINDS has developed, which really go across all diseases covered by this institute. And finally, we always want to support innovation, support technologies, support new findings that are going to tell us more about the human biology of Huntington's disease, but also help us to look for those targets that are going to be the most effective in terms of therapeutic development. So with that in mind, we'll focus on data and resource sharing. In terms of data sharing, one of the instruments we have at NIH is a database called dbGap, and it's a database of genotypes and phenotypes that was set up to help share both the genotype and phenotype data associated with genetic studies. Through dbGap, we've been able to transition data from the PREDICT-HD study. PREDICT-HD is a longitudinal study of HD participants who are examined before motor onset. And so this study started back in 2001, and there's a large amount of clinical assessments, genetic information, and imaging information that goes along with these participants. And so to bring that data into dbGap has enabled in a controlled way to share that data with the broader research community. So again, this just emphasized for PREDICT-HD, the phenotype data is shared through dbGap. The genotype data, which involves a genome-wide association study, is shared through dbGap. And PREDICT has also, along the time course of the study, collected biospecimens which are available through the NINDS repository at Cornell. In terms of partnering, which again I mentioned, it's been important to really find out shared interests and bring groups together to help accelerate research in Huntington's disease. And we've done that through partnering with the Huntington's Disease Society of America, the California Institute for Regenerative Medicine, CHDI, and the Hereditary Disease Foundation. Really working with these groups has helped to advance what we do for Huntington's disease research. Finally, in terms of supporting innovation, these are just a few examples of some of the recent advances and tools and technologies that are available for Huntington's disease research. So for instance, through ERA, we pulled together an induced pluripotent stem cell consortium, which is focused on development of induced pluripotent stem cell resources for both basic science and drug discovery. Through a collaboration with CHDI, we've focused our interest on target validation, increasing the information and scientific rigor around targets that could move forward for therapy development. And then in really transforming what we know about Huntington's disease in terms of genetics, we're supporting innovation that will help to identify genetic risk alleles and rare variants contributing to phenotypic modifications. And I'll give you an example of why, even though we know what the mutation is, that expansion of that CAG repeat region, there are still factors outside that contribute to variations in the phenotype you see when Huntington's disease is presented. To talk about data sharing and what PREDICT-HD is, both NINDS and CHDI have funded this study from 2001 to 2014. The study is scheduled to end in August of 2014. As I mentioned, it's a longitudinal observational study of individuals who are CAG, have that expanded CAG repeat, but have not actually manifest, at least in terms of motor manifestation of the disease. It's involved greater than 1,400 individuals, 63% female, 37% male. The average baseline age for the participants is around 41. It's involved 32 sites. And if I haven't mentioned this, the important aspect of Huntington's disease is that it's a rare disease. So it takes a number of clinical sites to identify the participants we've had in PREDICT-HD. Cases are basically the expansion positive people, and controls are the expansion negative people. Both the normal and the expanded allele are lab verified, and to date, over 200 participants have received diagnosis. So they've transitioned from that prodromal phase to diagnosis of Huntington's disease. Since this study has been going on over 10 years, there's 8 million phenotypic data points in the database. And as I mentioned, they include imaging, cognitive, motor, psychiatric, daily functioning, clinical assessments, as well as the collection of biosamples. So this gives you an idea sort of the breakdown of the cases you can find in PREDICT-HD. On the left-hand side, when I'm talking about status, and it refers to low, medium, and high. Low, medium, high refers to a CAP score. And the CAP score is a CAG age product index, which basically predicts how close you are to onset. So if you have a high CAP score, then you're close to onset of motor manifestation. And this basically gives you an idea of the percentage of the different stages of prodromal HD in the PREDICT-HD database. I think the strength, definitely one of the strengths of the PREDICT-HD data is the fact that the individuals were clinically examined once a year, and that clinical examination included MRI imaging. And so the study started out using most sites using a 1.5 Tesla, and by the time they transitioned into PREDICT 2.0, they were using the 3.0 Tesla imaging. And so that data and the measurements from cortical and striatal volumes are captured in the dbGap database. The other sort of summary of what's available through dbGap is given in this slide here. And basically, it tells you the cognitive, demographics, motor, psychiatric, functional, and genetics information you can find in dbGap, and whether it's a baseline. B stands for baseline, F stands for follow-up. In the 2.0 version of PREDICT-HD, it's a baseline, initial clinical assessment in 2.0, and then the follow-up. So it shows you that there is longitudinal data available, and it also shows you who was involved in the collection of that data. So given that we have this incredible resource, which is now broadly shared with the research community, we're interested in asking or finding out more about why there's variation in the age of onset for Huntington's patients. And the graph on the left-hand side gives you an idea that although you can track the age of onset with the CAG repeat length, the variation is considerable along that line. And so I think that's what's surprising, is that even after controlling for the effect of allele length, results indicate that fully 59% of the variability in residual age of onset is familial and attributable to either genetic or shared environmental factors. And so this gives you an idea where in the middle where you see that bar and equals 45, that's the age of onset. Sorry, that's the CAG repeat length. What it indicates as you go down is that a CAG repeat length of 45 can have an age of onset of 60, an age of onset of 40, or an age of onset at 20. So what contributes to that variation of age of onset is really what we're interested in finding out, and the focus is on looking for genetic modifiers. So there's several ways to go about this, and obviously, one of the initial studies is applying genome-wide association studies to look for risk alleles that may be associated with differences in age of onset. Because we have pedigrees and we have families where we can identify affected individuals across generations, we also have the ability to do family sequencing. And since we have a very extensive clinical assessment in terms of age of onset and variation in that, we can also do population-based sequencing. And so just, key factors in terms of family sequencing, knowing common versus rare variants, the accuracy rates of the sequencing and signal versus noise issue, genetic phasing in terms of the contribution of parental versus maternal, SNPs, and then the stratification of the disease and that family and how that is affected by family. So one example of taking this family sequencing approach is applying either whole exome or whole genome sequencing to identify modifiers of age of onset. So in this case, and in this study, the eligibility basically, it's defined where a family has a minimum of 4 members spanning 2 generations, where at least one symptomatic individual is present in each generation. And to coordinate, if those families don't exist right now and they are being collected, then we want to coordinate the clinical assessments being done with what's been done in PREDICT-HD. So in identifying these families, shared clinical assessment tools like the MoCA, verbal fluency, Stroop interference test, UHDRS are collected on the individual at the clinical visit, and then a skin biopsy and blood samples are taken to enable both collection of DNA, but also potential development of induced pluripotent stem cells. And so here's an example of what we see in terms of clinical variation in phenotypes in an HD family. So the first generation, in this case, the circle represents, a red circle represents an affected female, a red square represents an affected male. And you can see that in the first generation, you have 4 affected individuals really, where the age of onset is between 60 and 70 years of age. If we look at the second generation, you can see that again, and I should also mention that we know what that CAG expansion is. So in the second generation, you can see that on the left-hand side where we're looking at that red square, the male has a repeat length of 41, the female has a repeat length of 40. For the male with the repeat length of 41, the age of onset was 60, but in the female with a repeat length of 40, there was no evidence of Huntington's up to 70 years of age. That was also true if you look on the right-hand side of this pedigree. You can see that again, a female with a repeat length of 40 had no onset as of 70 years of age, whereas the male, that red square next to the female, had a repeat length of 41 but an age of onset of 56. I think probably the most dramatic happens in the third generation, where in this case it's a female who has a repeat length of 46 but an age of onset of 30. So using these pedigrees, these well-defined pedigrees with clinical assessments we can basically start to look for modifier genes. And to do this, again, just to emphasize that it's really data sharing of large data sets through this controlled access database like dbGap, and creating opportunities for new approaches to data analysis. So over the last year, NINDS has funded several studies to really help advance this modifier identification. So, for instance, the first one, genetic modifiers of Huntington's disease, was a study we funded to do a genome-wide association study using the Illumina Human Omni 2.5 platform. That genotype data was deposited with dbGap along with the phenotype data, and it's available to researchers to start looking at what modifiers might be through this type of approach. Another really interesting approach is the use of the data set that was developed from the MRI imaging plus the genetics information that was developed through the genome-wide association study, and combining those two in an imaging and genetic study that will hopefully tell us what SNPs are associated with changes in imaging. And so Vince Calhoun and Jessica Turner were funded recently to start looking at that type of investigation. Genetic modifiers of PredictHD phenotypes focuses on whole exome sequencing and looking at those individuals that have extended or sort of extreme phenotypes, again, to look for modifiers using a whole exome approach. And finally, the example I gave in the last slide is a family-based whole genome sequencing approach to identify modifiers where we have several generations, where each generation has an affected individual and a difference in age of onset. So again, taking these new genetic approaches to Huntington's disease will hopefully tell us more about how we understand variations in the phenotype. This broad data sharing has also helped to bring about new imaging technologies, application of technologies to Huntington's disease, as well as taking the existing imaging data set and applying new analytical tools to help us understand more about what's happening during the progression of the disease in the prodromal state. And I think that's one of the most significant findings from the PredictHD study, TrackHD, is that in that pre-manifest or prodromal state, there are significant changes in brain volume. And so understanding how that progresses and what it can tell us once we start developing therapeutics that could potentially move into that preclinical or prodromal patient population, it could be a huge tool for looking at therapeutic response. So examples of what we're funding just over the last 2 years, we're looking at the application of functional MRI in pre-manifest Huntington's disease. Steve Rao is the PI for that study. And then the next 2 studies are really using that existing data set, longitudinal data set for imaging that I mentioned, and looking at, for instance, how basal ganglia shape analysis and circuitry can help us define better what that prodromal state is and how it progresses. And the 4D shape analysis is really basically making individual movies of the changes in brain volume over time so that you can start to predict when changes in brain volume will be an indicator of progression to manifest HD. And finally, the last study is again using a new tool. We're applying it to Huntington's disease, where we're using FDG PET imaging to try and develop a clinical signature for Huntington's disease, where we're looking at changes in brain activity. So again, because we have an extensive clinical data set that includes imaging, we're able to share this broadly with the research community and bring in new approaches to analysis and help our understanding of Huntington's disease. The other thing I mentioned about PredictHD is that the study has collected biospecimens yearly from participants, and these biospecimens were collected in a standardized way, and they're available through the NINDS repository at Corel. Corel's pulled together a reference card for investigators to help them understand what the different samples are. This is an example of the reference card you see on the screen now. And basically, investigators interested in biomarker discovery can go to the NINDS repository and apply for access to those samples to be used in discovery projects. So here's an example, or several examples, of what NINDS has funded recently in terms of biomarker discovery projects using these valuable biospecimen resources from PredictHD. Clemens Schertzer is involved in the next generation of biomarkers. In this case, he's looking at changes in messenger RNA and small RNAs to see if you can develop a signature for Huntington's disease out of CSF. We're also using CSF that's been collected to look at proteomic markers for Huntington's disease and see if those proteomic markers can either help stage progression of the disease or tell us, how close to onset manifestation of the motor symptom a patient is. And so what I should mention is that over the last 2 years, not only has the PredictHD study collected plasma, serum, and DNA, but it also, as you can see, has started collecting RNA and cerebrospinal fluid, which helps in the 2 projects I just mentioned. The other project really is focused on a CSF-enhanced aggregation biomarker, and again, it's looking at can CSF from prodromal HD patients tell us something about the stage of the disease, and this is a functional analysis of what might be in the CSF that is necessary for aggregation of the HD protein. So when I talk, so we've talked about data sharing, we've talked about how that can drive innovation. The other thing that helps to drive innovation is partnering across organizations interested in advancing Huntington's disease research and looking for therapeutics for HD to develop resources that will help the community. And so in 2009, through the American Recovery and Reinvestment Act, NINDS funded 3 consortia to develop induced pluripotent stem cell resources. And in 2002, that was continued through another grant mechanism called the Cooperative Agreement. Basically, that opportunity, the discovery in 2006 that you could take a skin cell and apply transcription factors that basically drove that skin cell back to a pluripotent state that could enable it to become any type of cell in the body. That technology was what was captured under the 2009 American Recovery and Reinvestment Act and is the basis for the induced pluripotent stem cell resource that's available through the NINDS repository. So the concept of these consortia were to identify leaders in the fields of stem cell and neurodegenerative disease, clinical and cellular biology. So we needed experts in both fields to understand the biology of the disease, but also the biology of that induced pluripotent stem cell and how we could harness it to develop tools for both understanding biology and therapy development. The idea then, by bringing these groups of researchers together, was to create teams that could drive the science and work outside of the confines of an institutional setting. So the most of the investigators in these consortia came from a number of institutes, and by building this team effort, we brought people who may not normally work together. We created milestones to drive the research, so it was a very tight time frame, and it built on existing infrastructure that we had in NINDS, and in this case it built on the NINDS repository at Cornell. I think the most important thing in this consortium concept was to engage industry, because we really needed to work with industry to understand how these new tools might be useful in drug discovery efforts. So the investment under the U24, which was the second phase of funding, was $6.6 million for the 3 consortia over 2 years. And we were able to achieve this through partnering with non-government organizations in the California Institute of Regenerative Medicine. So it was a very important partnership in terms of really moving this effort forward. So in terms of resource sharing in human biology, again, I'm focusing on creating global research tools for advancement in Huntington's disease research. And again, this sharing is through our NINDS repository at Cornell. To date, that gives you an idea of, by building these resources, we've been able to distribute now over 700 fibroblast lines worldwide and 240 iPSC lines. And the distribution has been to both academic and industry investigators. So we made sure that when we were consenting participants to donate skin biopsies, that consent allowed for broad sharing of that resource. So this gives you an idea of, for instance, how you, if you're a researcher and you wanted to find out more about, in this case, fibroblast lines that are available for Huntington's disease, you could look at a family. And in this case, you can see at the top, the male is 54 years old and has Huntington's disease. Obviously, the wife is asymptomatic, basically gene negative. And then you have, in the next generation, 2 daughters who are gene positive and at risk but have not manifest Huntington's as yet. So those fibroblasts are available, and you can use those fibroblasts to basically reprogram and develop induced pluripotent stem cell resources. The interesting thing in that distribution I showed you, the first iPSC lines in the NINDS repository were available on September 30th, 2011. And so since that date, being able to distribute over 240 iPSC lines has been a huge advancement both for academic and industry investigators. Currently, we have 17 ALS lines, 18 Huntington induced pluripotent stem cell lines, 20 Parkinson's disease lines, and 6 iPSC lines for frontotemporal degeneration. This gives you an idea of what a page would look like and tells you about the induced pluripotent stem cells for Huntington's disease. We re-verify the CAG repeat length for the line, and we also, on that far right-hand side, provide a COA, which is a certificate of analysis, which tells you how the line performs, what to expect, what the karyotype is, and also protocols for thawing the lines and growing the lines. So through this consortium approach and development of induced pluripotent stem cell resources, several papers have come out from the consortia, from the HD-iPSC consortia. One paper focused on easy spheres, which were a stable and expandable culture system for generation of that pre-rosette multipotent stem cell. It enabled the consortium to readily ship these cells worldwide to share with collaborators. In 2012, this is a review from papers that were published in Cell Stem Cell, basically, and focused on the fact that these landmark papers on Huntington's disease, induced pluripotent stem cell consortia, basically demonstrated the establishment and characterization of a panel of induced pluripotent stem cell lines from HD patients, and more importantly, the successful modeling of HD in vitro. I mean, the fact that in a dish for a late onset neurodegenerative disease, you can see a phenotype related to the CAG repeat length was a huge step forward for using these types of resources in both understanding biology and in therapy development. So the paper was published in 2012, as I said, in Cell Stem Cell, and I think, like we saw with the genetics paper back in 1993, the authorship was the HD-iPSC consortium, and the contributors or collaborators were 58 in all. And so this collaborative approach to really accelerating both advancing resources and characterizing those resources has been huge for the field. And this gives you an example of the type of data that was published in that paper. So on the left-hand side, you see the characterization of the induced pluripotent stem cell lines. There was a 180 repeat line, a 60 repeat line, and a 33 CAG repeat line, which was the control line. When you look at these cells and characterize the survival or the basically how fast the cells die. The graph in the middle demonstrates that if you have a CAG repeat of 180, that has a much faster death rate than a cell line that has a CAG repeat of 30. So the 180 line is in orange and the 30 CAG repeat 30 line is in blue. What this consortium also demonstrated was that these lines are sensitive to BDNF withdrawal. So growth factor withdrawal, which is known to play a role in Huntington's disease, has an impact on survival. And so without BDNF, the survival rate drops both for the 180 and the 30 repeat line. But the bigger effect is obviously in the CAG 180 repeat. And the graph, or the bar graph on the right-hand side, basically shows you that, again, we're looking on the left-hand side, we're looking at in the white boxes, we're looking at the CAG repeat 30 lines with and without BDNF, and then 109 in the middle, and then a 180 on the right-hand side, where with BDNF you see certain rate of, in this case, they're measuring condensed nuclei, which is an indicator of cell death, and how that increases significantly when you withdraw the BDNF. So through this approach, basically, the consortia were able to develop a supply of patient fibroblasts, common techniques to modify the lines, and a uniform process for scaling and banking with a standardized quality assessment. So all of the lines that you see in the NINDS repository have gone through a standard QC process, which gives you some confidence in terms of the material you're starting with. In terms of induced pluripotent stem cell differentiation, what the consortia have demonstrated is that now you can get a reproducible lot-to-lot differentiation. You can apply uniform tissue culture practices and basically demonstrate that there's an ability to generate a large number of differentiated cells to support a set of interrelated experiments. And I think one of the nice things that has happened with the HD-IPS consortium is they developed a experimental protocol where one lab grew up a set of cells and then pelleted them and sent the pellets to different labs across the US to do transcriptomics and proteomics, and then bring that data back together to help develop a systems approach to looking at the phenotype associated with IPS cells carrying different CAG repeat lengths. And finally, and probably one of the most important things, is that the iPSC consortia was able to identify phenotypes, and phenotypes that are robust and reproducible in disease-relevant cell types, phenotypes in well-defined cell populations, and phenotypes that are consistent across the majority of the cell populations. And obviously, this is ongoing in terms of continuing to show the robustness of the phenotype, the reproducibility of the phenotype across labs, and also to develop assays that are easy and portable between labs. So really, that's where technology is headed, and it's been an amazing process in the fact that it's happened over a very short time period. So again, to give you an idea of the distribution of the induced pluripotent cell lines by disease in the NINDS repository, you can see the bar graph on the right-hand side. It highlights mutations, for instance, FIG4 and SOD1 associated with ALS. The Huntington's mutations are in turquoise blue. Synuclein, LRT2, GBA, and PARC are mutations associated with Parkinson's disease. It gives you an idea of the number of lines of each mutation that have been distributed to date. Also, the pie graph shows that 60% of the iPSC line distribution has been to academic labs, and 40% of the lines have been distributed to industry. And again, this gives you a breakdown in terms of, the other pie graph gives you a breakdown in terms of the number of lines that have been distributed based on disease: Huntington's, Parkinson's, ALS, and controls. So again, we've developed these induced pluripotent stem cell resources for both understanding basic biology and hopefully advancing therapeutic development in Huntington's disease. But I also want to emphasize that the Huntington community has developed a number of mouse models of Huntington's disease. And this paper that was published in Neuron in 2011 gives a really nice example of the types of mouse models available. They're based on transgenics, expression of exon 1 fragment, full-length fragments, yak and back transgenics, and also knock-in models. And so these investigators have basically given you an outline of when to expect certain phenotypes in these different cell lines over a 2-year period. And the phenotypes most commonly looked at in these mouse models are focused on motor symptoms based on rotor rod, neuropathology like intranuclear inclusions, excitotoxicity where you're measuring electrophysiological differences when you have an expanded CAG repeat, transcriptional dysregulation driven by the CAG repeat expansion, changes in mitochondrial function, in this case calcium buffering, and then again looking at network effect in terms of LPD and LTD changes when you have that CAG repeat expansion expressed. So very nice resources. There's at least 13 mouse models commonly used in the field right now to help us understand the disease biology and the basic biology of Huntington. So this is an example of what we know about Huntington's disease in terms of bioenergetics. There have been a number of publications over the last several years that have focused on PGC1 alpha, which is a PPAR gamma coactivator 1A alpha. It's a coactivator of nuclear receptor genes, and so because of that, PGC1 alpha plays a significant role in mitochondrial biogenesis and oxidative phosphorylation. So we know that mutant Huntington can impact the ability of PGC1 alpha to interact with the nuclear receptor. And when it inhibits that interaction, it basically changes the transcription or the ability to work with the nuclear receptor at the promoter site and changes the transcription level of those target genes. And so that interaction of PGC1 alpha mutant Huntington brought together the fact of what we know in human biology of Huntington's disease, that there is a dysregulation in the transcriptome of cells expressing, neurons expressing Huntington protein, mutant Huntington protein, and there's also a change in the bioenergetics or mitochondrial function in those cells. And so this was a link between the mitochondrial dysfunction and the transcriptional dysregulation. The interesting thing is that not only is PGC1 alpha expressed in neurons, but it's also expressed in oligodendrocytes and has an impact on expression of myelin basic protein. So as I mentioned, several labs over the years have been looking at PGC1 alpha and how that impacts mitochondrial function in Huntington's disease. Another major advancement really focusing on an omics approach to understanding the Huntington protein, both the mutant and wild type protein, is understanding what that proteomic interactome is for Huntington's disease. And so William Yang's lab published this paper in Neuron in 2012, which took a omics approach, basically immunoprecipitated Huntington protein from different brain regions of BAC-HD mice at 2 months and 12 months of age, and looked at what proteins mutant and wild-type Huntington interact with. And they built these networks based on function. And the one I'm going to focus on in this red box is the network that includes the Huntington protein itself. And so basically this network defines proteins involved in chaperone and protein clearance, and other known interacting proteins that have an impact on Huntington function. And taking this approach has really broadened what we know about Huntington's disease and the proteins it interacts with and the pathways we should be looking at. This is an example where this is a specific case of a protein that mutant Huntington interacts with, and that interaction impacts synaptic function. So in this case, normally in an adult synapse, NMDA receptors are tetramers and made up of either 2B or 2A subunits. In development, there's a 3A subunit that basically allows less calcium to enter the cell. As the synapse matures, those 3A containing tetramers are removed from the synapse by a protein called paxin-1. And that paxin-1 basically helps that clathrin-coated clearance of those 3A receptor containing, NMDA receptor containing molecules so that the synapse is expressing mostly those tetramers that can allow high entry of calcium into the cell. When mutant Huntington is present, mutant Huntington interacts with that paxin-1 protein and inhibits the ability of the protein then to pull those 3A containing receptors from the cell surface. And so you end up with a dysfunctional cell in that it's expressing receptors that only allow low calcium entry, and that leads to basically synaptic dysfunction that we've seen in Huntington's disease. And so this is also a paper talking about the easing of the NMDA receptor traffic jam, and the seminal paper that explained how that 3A subunit gets trapped at the surface and isn't cleared as it normally would be in the mature synapse. So the other thing we know about Huntington's disease is that there's differential neuronal vulnerability. And there's several mechanisms that can drive this. For instance, when you have a cell, a neuron that's aging, it becomes sensitive to oxidative base oxidation, basically, where there's a mechanism within the cell called base excision repair that brings in this glycolase enzyme, OGG1, to repair the excision that's made because of that oxidative base. Unfortunately, the polymerase will come in that excision area and create a hairpin loop that results in strand displacement and slippage. And through this strand displacement and slippage, you can actually get an expanded CAG repeat. And so that is an instance where we're explaining how a toxic oxidative cycle in neurons that are vulnerable can result in a neuron or a cell-specific expansion of that CAG repeat. The other thing we know is that the proteins that help to stabilize that hairpin, basically MESH3 and MESH2, are also influenced in terms of having polymorphisms in their promoter regions that will allow more or less protein being made. If less, for instance, MESH3 is made, then again, that contributes to the inability to repair these cleavages that are brought about by the oxidative cycle. And those are recent discoveries that have been published over the last couple of years. The other example of where you can demonstrate neuronal vulnerability is a methodology that was published just in 2013 describing the use of dendro-2, which is a photoswitchable fluorescent protein. And so you can use basically optical activation to do a pulse chase experiment and ask what is the half-life of the wild type versus mutant Huntington protein. And so this work was done by Steve Finkbinder's lab. And you can see that through pulse chase experiments, the wild type Huntington protein is more stable than the mutant Huntington protein. And that, again, is reflected here as well. When you look at our cortical neurons more susceptible than striatal neurons, what you find is that striatal neurons, which are shown in red on the left-hand side in the red boxes, are more susceptible to death due to expression of that expanded CAG repeat than cortical neurons are. And basically using this dendro-2 optical imaging approach, this lab demonstrated that it's not the amount of protein, but rather the half-life of the protein that determines the susceptibility of the neuron, the neuronal type, to mutant Huntington. And so if you have a longer half-life of mutant Huntington, it indicates that that neuron's capacity to clear the protein is not as effective, and that's what's contributing to the cell death. Finally, I just wanted to emphasize again that partnering and innovation has really driven what we know about target validation. And so these are examples. This was an example of a grant mechanism that we've had over the last 3 years that funded 12 grants to really further the clinical or preclinical evidence around specific targets. And you can see the targets listed there. So I'm running out of time. I just wanted to highlight that one of the grants that was funded really focused on understanding how phosphorylation of two serines in the N-terminal region of the Huntington protein can impact the localization of the protein, the aggregation of the protein, and the toxicity of the protein. And so this was seminal work in terms of understanding how post-translational modifications modify the toxicity of that CAG expanded repeat. And this work was done by a number of labs and has really had an impact on the field over the last couple of years. So finally, I want to emphasize where target validation and therapy development is headed. And one of the key tools that has been used recently is looking and focusing on the Huntington messenger RNA and protein itself as the target. And so one way to develop a therapy around that is to use antisense oligonucleotide therapies that are targeting the mutant Huntington or the Huntington protein. And so you add the antisense oligonucleotide, it binds to the messenger RNA and creates a DNA-RNA hybrid. And then RNase H preferentially removes that RNA from that DNA-RNA duplex, releases the oligonucleotide to, again, go back and identify messenger RNAs, specific messenger RNAs. And by using this approach, you can basically develop what they call a Huntington holiday effect, where you're lowering the amount of mutant Huntington protein that's produced because you're removing from the cell that messenger RNA associated with it. And that is a therapy that has a lot of potential, and it's been rapidly being advanced by labs like Don Cleveland, working in collaboration with ICES to find that the best clinical candidate to test in early phase clinical trial. And this just gives you a little more information on the data around that experiment, where again you can see on the left-hand side of the screen in a back HD model that only the human Huntington protein is reduced when you're using an antisense RNA directed directly at that sequence. So finally, I've mentioned what NINDS funds in terms of Huntington disease research. There are other opportunities, if investigators are listening, for potential programs that can be applied for Huntington disease research. And the example is recently the extracellular RNA communication program funded through the Common Fund funded a Huntington disease project that's using or developing exosomes as delivery systems for siRNA to, again, use a gene silencing approach to as a potential therapeutic moving forward. Also, the nanomedicine project funded through the Common Fund is looking at how chaperone proteins may play a role in helping to clear mutant Huntington protein. And again, that's was a funding opportunity through the Common Fund. So I encourage investigators to look not just within NINDS, but across NIH for potential opportunities, funding opportunities that can be applied to Huntington's disease. Finally, there are tools being developed and programs in place that really provide incredible information for understanding human biology. And so the NIH Blueprint projects, and an example of this is the Human Connectome, provides a very rich data set, 60 healthy individuals where diffusion MRI, resting state fMRI, and other imaging modalities have been used and are available in the data set to be shared with the broader research community. So again, look for those data sets that will help advance Huntington research. And finally, another Blueprint project. NIH Blueprint projects are a result of bringing together neuroscience institutes focusing on areas of interest that cross the neuroscience institutes. So in this case, the NIH Blueprint project on neurotherapeutics provides an opportunity to fill in that valley of death. And you can see it's highlighted in purple in this slide. And it's basically moves from hit compound to a phase 1 clinical study. And anywhere along that pathway, a candidate can enter and move and use the resources available to help move a clinical candidate or a preclinical candidate forward. And so this gives you an idea of some of the projects that are funded under the NIH Blueprint program. You should look in this space. It's anticipated that another funding announcement will be available. And for NINDS, this will be the program that will be used to advance any small molecules towards an early phase clinical trial. So again, make sure you look at this space and look for funding opportunities within the neurotherapeutics Blueprint program to help your project move forward. And really, that's all I have. And if you have any questions, I hope I've sort of given you a broad idea of what's funded in NINDS. We're excited about Huntington's disease research, major advances that have happened, and funding opportunities both within NINDS and outside in the broader NIH space for Huntington's disease research. And I can take a couple of questions. And so the one question I have is, would you be able to provide info for HD patients interested in participating in research or clinical trials? And yes, we can definitely do that. One place to find that type of information is in clinicaltrials.gov. And if you go to that website, you can basically put in Huntington's disease, and it will give you a list of the clinical studies currently active and enrolling participants. You might also be interested in Enroll HD, which is sponsored by CHDI. And again, it's a longitudinal study that's taking all HD participants, both early stage, prodromal HD, and manifest HD, and does a clinical assessment each year. And that's a project that individuals might be interested in finding out more about. And so to find out more about that, I would go to the CHDI site and look for Enroll HD. Okay, with that, thanks for listening, and I hope I've been able to at least inform you on the National Institute of Neurological Disorders and Stroke, our strategy on Huntington's disease, and what you can find at NIH. Thanks again.